Alright everyone, so welcome to the third lecture for our week 3, very important, on neurochemicals, neuromodulators, and neurohormones. So overview, we'll just talk about this thing first, and followed by how these different kinds of drugs, right, different kinds of neurochemicals work, and then basic principles of drug effects. You guys will like it, and then the last part will be really for tutorial 2. We absolutely do not have the time for the last part. The last part is not so crucial, but then it's really for knowledge building. Right. So first thing first, okay, what are neurotransmitters? That one I talked about briefly last week. However, I did not identify, right? I did not identify during week 2 what these neurotransmitters are. So, but basically, remember the second bullet point is very important. So they participate directly in synapses by acting on neurons in the immediate vicinity. So the whole keyword here is directly, the key adverb, right? And that thing we call it synaptic cleft, right? Just for the sake of clarity, the one that you're looking at right now, on the right panel, that is called the synaptic cleft. I mean, the synapse itself can be much wider, but this small little junction is called a cleft. And neuromodulators is a bit different, and neurohormones, right? So the keyword here is indirectly. They participate indirectly in synapses by acting on more distant neurons. And I will show you what I mean by that in the next slide. So after they communicated with the target cells, normally the neuromodulators will diffuse away from the point of release. Similarly for neuropeptides. Okay, but neuropeptides is more like in the blood capillaries, in the bloodstream. Always remember that. And so the last point is that neurohormones are produced by our limbic system, hypothalamus, adrenal glands. Where are the adrenal glands? On top of our kidneys, right? Attached to our kidneys. And our enteric nervous system, right? They travel in the blood supply to reach their final targets. So these are two very nice figures. Two very nice figures. So first thing first, right? Neurohormone. So what it does is that it travels through our bloodstream, our blood supply. And the second point is very interesting. Sometimes it can behave like a neuromodulator, right? If the target cell have a specialized receptor cell. That means they have a special recognition site for the neuropeptide to latch onto. And then the right panel, that is what we call neuromodulation for neuromodulator. And the keyword here is that they are often, not always, released from very cities. Very hard word to remember, okay, and it's quite hard to pronounce. Okay. So they are a little bit like swellings, and enlarged, I would say like enlarged places along unmyelinated axons. So another bit of information to remember. So they're not myelinated, the one in the picture is not myelinated, no Schwann cells. And they diffuse away from release site to influence activity on both pre and pre-synaptic terminals. And they're slower, but they have long-lasting effects. Right? How long is it? Okay, it can be a few minutes, a few hours, and even days. Yeah. Okay, right. Identifying neurochemicals. So a neurochemical, in essence, it covers everything. The one that we talk about, neuromodulator, neurohormone, or neurotransmitters. So ultimately, they're just chemical substances. They are released by one cell, and they produce some response reaction in the target cell. Simple as that. And there are just 3 principles that we generally stick to. So a neurochemical must be present within a presynaptic cell. Number 2 is that it must be released in response to presynaptic depolarization. That one I need everybody to revise with 2 materials. Number 3 is that it interacts with specific or specialized receptors on the postsynaptic cell. Okay, number 3 is a bit too much, I know. We'll talk about it in a few in the slides that follows. This like types of neurochemical, right? This entire decision tree-like diagram is taken from your chapter 4. Okay, chapter 4, very important. I will put an asterisk there, okay, even though I didn't. Right, so put an asterisk there. Remember, okay. So what are the neuromodulators? So we have all this in small molecules, right? 4 subclasses apart from the amino acids, right? Which I normally just refer to as AA. So the AAs are not. The AAs are neurotransmitters, amino acid neurotransmitters. Remember. Okay, neuropeptides, very special. They can behave as both neurohormones and neuromodulators. Wow, this one I have to revise. I didn't know this. Okay, I didn't know this. And the last part is gastrotransmitters. So that comes from nitric oxide, very common in our bloodstream. We actually synthesize such gases right, from all the biochemical reactions in our body. And then there's also carbon monoxide that we can inhale in, right, and kill ourselves. I'm not going into that. Right. So this is a very important table comparing our small molecules with neuropeptides. I'm just going to read on a few important points. Right. So synthesis, small molecules can happen anywhere in a neuron. And neuropeptides, it happens, they're synthesized in the cell body, and they need transport to the axon terminal. Recycling of vesicles, small molecules, yes. Neuropeptides, no. Activation part is very crucial. So the small molecules will generate moderate action potential, and neuropeptides surprisingly can generate very higher action potential. So what we learned in week 2, we are talking about that is moderate action potential. All right, remember that. Okay. And deactivation, we're talking about reuptake or enzymatic degradation. So in the synaptic cleft, right? That's very, very common. I talk about it so many times. So it's in tutorial 2 we covered it as well. Degradation in the synaptic cleft. And neuropeptide is no reuptake because it's in our blood supply, and it diffuses away from the synapse or enzymatic degradation. It can be degraded as well, right? Right, so first thing first, the first neurochemical we're going to talk about, acetylcholine, right? ACH. Remember the first point, right? It is the very first chemical messenger, and it is prevalent in our central nervous system, right? In our brain, in our autonomic nervous system. Right, autonomic nervous system, in my previous lecture, what I talked about, right? It is sending all kinds of signals, right, to our muscles. Yeah. So very important. Yeah. So neuromuscular junctions, remember that. Okay, it's receiving all the signals. And what happens when you have excess of ACH is that you will have hyperactivity or rigidity of muscles. Yeah. So later on we'll talk about a drug that leads to such excess ACH. And when it gets reduced, you will have muscle weakness and flaccid paralysis. So it's really like the side of your face becomes very, very soft and becomes very droopy. Right? Literally, you see the skin, basically your skin is not tight. Yeah, it's quite a very hard, very difficult condition to have. Not good for young people like us. Right? And if it gets more severe, it can affect cognitive functions, can lead to cognitive decline. Yeah. Right. So we have 2 types of cholinergic receptors. They're also called, it's also possible to simply write them as ACH receptors, right? So the name, the terminology is very unique, with regard to the fact that they are called in addition, right? So the name with respect to substances in addition to ACH, to which they also respond to, right? So we have nicotinic receptor, so it's ionotropic receptor, and basically it responds to both nicotine and ACH. Fast responses. This means that once it receives nicotine or ACH, the ion channel can be sodium channel, can be potassium channel, it will open directly. And this is present at our neuromuscular junction, central nervous system. Yeah. So nicotine, we all know what it's good for. Right? So tobacco, it increases arousal, attention, REM sleep. Later we'll talk about that rapid eye motion. Enhances learning and memory. So it's actually quite a good thing to have, right? And muscarinic receptors, and this is a slower kind of receptor. This is metabotropic. It's much more common in the central nervous system than nicotinic receptors, activated by both ACH and muscarine, which refers to this mushroom that we call Amanita muscaria. Yeah, this is a poisonous mushroom here. Okay. Right, very, very interesting fact to know, yeah. So this is just a diagram, the first neural pathway, or what we call a neural system, to take note of. So we had all these cholinergic neurons, widely distributed, right, in the brain. And just take note that they originate in our basal forebrain. I highlighted it here, basal forebrain, ventral prefrontal cortex, and our brainstem. Brainstem, yeah, it's just like a mic I'm holding here right now. Okay, and this is really like the top part of it, in the midbrain. Okay. We see sense projections, the limbic system. We talk about limbic system in our first tutorial, and with amygdala and hippocampus, right, the 2 main structures. Also like cerebral cortex. It also sends to lower half of the brainstem. So this is like the lower half of my mic. This is, brain-wise, right, it will be called the medulla oblongata. Okay, take note of that. So projections deteriorate with in persons with AD, Alzheimer's disease, unfortunately, right? Yeah. So that leads to motor dysfunctions for people with this disease. All right. Next, this is very important, right? We actually have a cluster of very important neurochemicals. So monoamines. So what are monoamines, right? So briefly, for those of you who still remember your chemistry, right, organic chemistry. So amino, right? So it's a combination between amino group and a singular amino group, one NH2 group, and we have something called a benzene ring, right, with OH group, and then we call it an aromatic ring. So it's a hydroxy aromatic benzene ring, in other words. So it's like that, right? If you Google for it, that's what we call monoamine. And catecholamine is a combination between catechol and the amino group. Right? So catechol is also another kind of benzene ring structure, if you look for it. Similarly for indolamine, but indolamine is like two benzene rings. Yeah? So they all have one amino group. So just take note of it. So catecholamine, very hard to pronounce, I know. Catecholamine. So they consist of dopamine, norepinephrine, and epinephrine. Right? Okay. Another two very difficult words to pronounce. Right? So Americans like to pronounce it as noradrenaline, norepinephrine as noradrenaline. So it's the same thing, exactly the same substance. And epinephrine is also called adrenaline. Yeah. So, and that gives rise to the phrase adrenaline rush. When you're on a high, doing sports, you're having adrenaline rush. Yeah. Histamine, right, important for wakefulness. I'll talk about it later. Indolamine, the most famous one is, of course, serotonin. Right? So in tutorial today, I talk about serotonin, very important for regulating our mood. Right? Not enough serotonin, you may have depression. That is based on the conventional theory. Okay. And also note this adjective biogenic. We make them in the brain, we make them ourselves, so it's natural occurring. And normally it's kind of uptaken from synaptic gap, or they can be broken down, right? Or small molecules can be broken down by all these enzymes. Monoamine oxidase is one, and then we have something called CMOT. Hard to pronounce, I'm not going to pronounce this, right? Catechol-methyl-transferase. I hope I got it correct. COMT. Right? Alright, so we come to this very crucial slide. So catecholamines share a common synthesis pathway. Everything starts, right? If you can't remember, everything starts with the letter T, right? And T stands for tyrosine, right? So tyrosine is a kind of amino acid, right? Just remember it's amino acid. And from there, we get a sequence of enzymatic reactions. So a series of enzymes with very long and difficult names, which I will not test anybody on. But you need to know the end product, right? The end product is epinephrine, adrenaline. Okay, and then the first thing to be produced is dopamine. Okay, circle it. Dopamine, put a 1 there. Number 2, norepinephrine, put a 2 there. Last one, epinephrine, as I said before. So the difference between norepinephrine and epinephrine is that norepinephrine is generated in the intracellular vesicles. It's made there. And epinephrine is generated in the intracellular fluid, and then is packaged into a vesicle. So there's a circle here on the right, the oval. You can see that Ne gets converted into E, and E gets put into a vesicle. Well, this is kind of interesting, right? This is the mechanism that epinephrine is being produced. So dopamine functions of dopaminergic systems in the brain. So later on, when we talk about psychoactive drugs, you will see that they're pretty much all dopamine agonists, right? So they really, dopamine gets really, really highly activated, excited, and it is, of course, the first that's generated amongst the three catecholamines, and it's highly associated, I would say highly associated with cognitive and motor control, motivated behavior, arousal, reward processing. The words like motivation and reward, these two words are almost always tied to dopamine. There's so much, many papers written on this that it's thousands of papers written on this, I would say. Yeah. And its altered activity with all this dopaminergic system, dopamine release will lead to Parkinson's disease, right? That's when your substantia nigra in your midbrain degenerates. You get Parkinson's disease. ADHD, actually, I'm not too sure, right? ADHD can happen along the pathway, along any kind of region along the pathway. We also have schizophrenia. I believe that schizophrenia is highly tied to our medial temporal region and our prefrontal cortex, the connections between them. And of course, with our midbrain. So ADHD, for a lot of you are writing on that. If you take note of this, when you get lower levels of dopamine, we had this drug called, retelling the generic name, to increase dopamine levels. Take note of it. And schizophrenia, we had this very, very famous drug called haloperidol. That kind of blocks the unblock effect of dopamine. So when you have a lot of dopamine in your synapses, you tend to have delusions, hallucinations, paranoia. You want to block it. So haloperidol is an antagonist, in other words. Yeah. All right. So dopaminergic systems in the brain continued. Take note. We have 3 major pathways in our brain. I'll go through them one by one. I want everybody to remember, especially the first one. Okay. If you cannot remember the second and the third one, remember the first one. So the first one is what we call the mesostriatal or the nigrostriatal. Yeah, the nigrostriatal is not so often used. Usually we call it the first. Yeah. So it goes from our midbrain, meso, right, substantia nigra to be precise, right, to the striatum, which is in our basal ganglia. So striatum last tutorial, if you still remember, right? Striatum is the combination of the caudate nucleus and the putamen. That we call it the striatum because they form stria. You can actually see stripes under the microscope. And of course, if your substantia nigra degenerates, get lesion, you will have Parkinson's disease. Okay. Then next pathway is the mesolimbic pathway. So linking our midbrain with the limbic system. So limbic system has a lot of structures. Specifically, we're talking about nucleus accumbens. Right, nucleus accumbens so small, right? You can see it here, so small, right? Very, very close to our basal forebrain. But it's highly involved in all this motivational, reward-related behaviors. That's why I spend so much time talking about this small region, because tons of research done on nucleus accumbens. Again, I mean, these 2 words come up: motivation and reward. Remember. So the last pathway, we call it mesocortical pathway, the easiest pathway to remember because I think this goes to our entire cortical system, right? Frontal lobe, right? Even the back part of it, okay. But primarily to the frontal lobe, right? The basal forebrain part, right? So it stretches from ETA ventral tegmentum area to prefrontal cortex. It regulates our executive functioning. Planning, for instance, like, I want to plan for the midterm, I'm activating this pathway. Alright, so with that, we move on to the noradrenergic system in the brain. So we talk about norepinephrine, noradrenaline, and we have, at the same time, we need to talk about the product that comes, right, from the action of a particular enzyme on norepinephrine that gives us epinephrine. So epinephrine is a product of norepinephrine. So this part I spent a long time making this slide just to get everything clear. It's very hard to get lost. Very, very easy to get lost if you read. Later on, I'll talk about the last bullet point, right? So always remember norepinephrine and epinephrine. These are 2 primary neurochemicals, catecholamines, of our sympathetic nervous system, which produces fight or flight responses, right, as I said last week. And our parasympathetic nervous system, right, is for rest and digest, relaxing our muscles, right, making us want to sleep. That is ACH, acetylcholine. Don't get them wrong. Don't get this fact wrong, right? And I also want to bring up the fact that it is initially, right, our midbrain secretes norepinephrine that travels down the spinal cord, right, the fiber along the spinal cord, and then it leads to all the internal organs. And once it reaches the kidney, we have adrenal glands on top of the kidney, and that will make the medulla, right, a particular spot in our adrenal gland to secrete adrenaline, epinephrine. So that's where we get this adrenaline rush. Yeah. At the same time, I do have to mention that our brain do secrete adrenaline, but you need to understand, right, in our brain, norepinephrine gets produced first. So norepinephrine also, the amount of norepinephrine in our brain is always larger than that of epinephrine. And then later on, as it reaches our internal organs, our stomach, our intestines, our kidney, right, we get more adrenaline rush, more adrenaline produced. Yeah. And the last point is just, we have all these receptors. This is just for your knowledge. I do not think it's actually in our textbook, but do take note of it. We have so many different subtypes. Receptors is not just one kind of receptor. Usually like a few, right? 1, 2, 3, 4, a lot basically. So we have alpha subtypes. Okay, number 1 is responsible for vascular smooth muscle functioning, and number 2, from the brain. That's in our brainstem. Yeah, so they're located in different regions. So beta, we also have 1 and 2 in heart and lungs. Both of them found in our respiratory system. Okay, with that, I just want to continue, right? This is the pathway, right? So norepinephrine, it originates from the pons, specifically what we call the locus coeruleus, the brain bridge, of our brainstem, and hypothalamus. Right? Hypothalamus, for those of you who still remember, hypothalamus is part of our limbic system. And locus coeruleus, again, is a very small nucleus, very small structure, right? And it goes all the way to the spinal cord, right? Prefrontal cortex, parts of the brain. And surprisingly, it only has 4,000 cells. Yeah, that's why it's so small, right? But they have, the axons have 1,000 connections each. Yeah, I cannot really imagine. Yeah, it's you draw on this brain. Yeah, it's kind of impossible. Yeah. So yeah, try to find a way to visualize this. It's amazing. So we'll talk about indolamine next. All right, this is not as important as the first 3 catecholamines, but still it's very important, especially if you choose to write on depression, right? So you have to write about this. Okay. So this is a bit unusual because it's synthesized from tryptophan, another T, okay, not tyrosine but tryptophan, right? It is an amino acid, right? We call it an alpha amino acid that we get from our dairy products. Yeah, it's non-biogenic, that is, we really have to eat it. If you don't eat such things, you may not even get enough serotonin. Yeah, the pathway itself, do not have to worry. Okay, we have an intermediate substance called 5-HTP. Don't have to worry. Yeah. Ultimately, you just need to know that it's made from tryptophan, and ultimately we get serotonin. So yeah, our enteric nervous system is involved. Yeah, so 95% of such neurons are found in our enteric nervous system. Number 3, I think I mentioned this a little bit, right? So because it's related to giving us a sense of happiness, right? Sense of calm. So of course it regulates our mood, sleep, appetite, and there's 15 subtypes. Okay, this is really for your information, non-examinable. Right? 15 subtypes. So all but 1 is metabotropic, right? So 14 metabotropic receptors, and many autoreceptors. So they really take it up. Have to take out the extra serotonin in the cleft, synaptic cleft. Yeah, so that's what they're involved in. All right, so this is, I think, the 3rd pathway to know, right? Serotonergic pathway, and we have this very unusual nuclei, right? All these spots. It is pronounced as Raphi nuclei. I spent a long time pronouncing this word. Right? In the past, I pronounced it as Raphi or Raf. That is just wrong. Okay, this is called Raphi. Yeah. Try pronouncing it right. Okay. And yeah, we have a lot of such nuclei, Raphi nuclei, along our brainstem. It projects to a lot of regions. Okay, our spinal cord, okay, all the way down spinal cord to our cerebellum, limbic system, and cerebral cortex. Yeah, maybe that's why it's so important, involving all this rest and digest functions. So control of mood, controls of aggression. Not enough serotonin, you may be aggressive. And if it's at a higher level, it will be highly crucial to regulate our learning and memory. And so that gives us to this serotonergic or serotonin theory, speculating on that. Such systems are affected in major depressive disorder. So you can look it up. Okay, there's some new evidence saying that no, it's not that crucial. Yeah, so just look at the URL for those of you who are working on depression, writing about depression, right? I'm sure this will be useful. So other monoamine, this is melatonin, very popular. I first came across this in my secondary school bio textbook. I still remember it, or I always remember it because the name is very easy to remember. So it's involved in sleep and waking. The source I have to remind. So it's from a pineal gland, right? Very hard to see, right? It's just somewhere posterior right? Somewhere close to the posterior commissure. That's our tutorial 2 slide. Yeah. Very hard to see, very hard to see. Yeah. And it's synthesized from serotonin. Yeah. Okay, that's all you need to know. Okay. And histamine, it's again, it's related to wakefulness. Its source, okay, this very difficult word, tuberomammillary nucleus, TMN, okay. And I think some of you can see it, right? So this is the TMN is very close again to our basal forebrain. I was always kind of imagine it as next to the nucleus accumbens. Yeah, it's extremely small. And it projects to our basal ganglia, which is just right anterior to it. And synthesized from this amino acid, L-histine, that's where it got its name, yeah? So the histine and amino group, it becomes histamine. So antihistamines, right, I'm sure a lot of you know this, so if you got a cut, got a fever, you take some Panadol, right, some Panadol will contain this antihistamines, right? Make you want to sleep, huh? As a way, for you to ward off the pain. Alright, so let's carry on. Yes, amino acids, finally, yeah, our amino acid neurotransmitters, extremely interesting, yeah. So first we talk about glutamate. Possibly, I mean, for all the neurotransmitters, I always put glutamate at the very top, yeah. So I had a paper on this, yeah. Not investigating glutamate itself, but the pathway involving glutamate. So it's the most excitatory neurotransmitter. And it's synthesized from this very complicated sounding metabolite called alpha-ketogluteric in our mitochondria. Yeah. And excess is, of course, harmful, it's toxic. And we have 2 different kinds, right? So this is AMPA, NMDA, these are ionotropic receptors. So they open up the ion channels once they receive the glutamate directly. And metabotropic involves an intermediary secondary messenger, the G protein, that is much slower. So because the G protein has to tell the ion channel to open, right? So this is what I mentioned in my tutorial this early morning. Right? So for future tutorials, tomorrow and on Wednesday, right? Take note of this slide, okay? Put an asterisk. This one, put an asterisk as well. Amino acid messengers, glutamate, right? So subtypes of ionotropic glutamate receptors, kinase and AMPA. I'm not going to pronounce the entire name of AMPA, it's just too long, right? It's from our textbook, right? Can look it up. So what happened is that, this is what I said, right? So glutamate molecule, as long as it binds with the receptor, the sodium channel will open directly, and sodium will enter the cell, right? So that leads to depolarization. So that's excitatory postsynaptic potential, slight depolarization, right? And NMDA, okay, so it's ligand dependent. It is also voltage dependent. Wow, I mean, this is amazing, okay? I want to go this thing a little bit slower. So it can be activated, right? But it requires sufficient depolarization of postsynaptic membrane and binding. So the first bullet point here, under NMDA, so at rest, right, the ion channels are blocked by magnesium ions. So what happens when you have depolarization? Right? Depolarization alone, right, it will eject the magnesium ions, right, and that will allow the sodium ions to enter. Right? So, and also note that a calcium ion is 2 times bigger than a sodium ion. So when you first open, the sodium ion will enter. It's 2 times. If you look at the mass number, it literally is 2 times. And once glutamate can bind with receptor, right, the channel will open more widely. Sodium and calcium ions will enter the neuron, and that leads to further depolarization of cell. Right, that will lead to an action potential, a stronger action potential, if need be. So take note of this. This is a very highly unique aspect of NMDA receptor. Put an asterisk in front of mine. All right. So next we go to GABA. So GABA stands for gamma aminobutyric acid. Okay, very long name. Right. I'm not going to the organic chemistry part. That one everybody can Google, right? It's not important. What you need to know about its mechanism, right? So the first thing that you must know is that it is the most common inhibitory neurotransmitter in the brain, in the CNS. And we synthesize it from glutamate, right? Enzyme, GAD, is for your information. So we have 3 types, okay? GABA A, B, C receptors. So A and C are ionotropic chloride channels. So it allows chloride ions to enter, and B is metabotropic potassium. So this is responsible for the repolarization phase. I would say A and C are also involved in the repolarization phase because you are allowing the chloride ions to enter. Yeah. However, this is really for everybody's general knowledge, right? So if you're asked to describe action potential, you don't have to mention all these things, right? It's really a bit too much. So the last point is hyperpolarization can occur whenever GABA A, GABA C allow chloride to enter the cell, similar to what I said earlier, and when GABA B allow potassium to leave the cell. Right? Everybody got that? Yeah. Think about it, okay? Think about it, right? Yeah. So I want to talk about very quickly about the metabolites. So this is related to the tutorial slide on caffeine. Very interesting, right? So ATP, I remember, it is not a neuromodulator. It is not a neurotransmitter. It is an intracellular metabolite. So when it gets decomposed, we have the byproduct called adenosine. So adenosine, it will make us relaxed. Right? So ATP, of course, is the energy molecule that our mitochondria produces. Right? So it is a metabolite, okay? Right, but it's still regarded, as far as our brain goes, right? So in the CNS, I will correct the end, right? And the connections with autonomic neurons and the vas deferens, our sperm duct, our bladder, our heart, and gut. Yeah. So ATP and adenosine are associated with pain perception and sleep-waking cycles. Yeah. Just think of the word sleep, right? And everything will pretty much make a lot of sense, right? So this slide, ATP and its byproduct, adenosine. So adenosine produced by the degradation of ATP, right? And if our brain is very, very active, we get to see a lot of degradation of such ATP. So adenosine levels will increase, right? And if it's high, right, you will trigger non-REM sleep. So that will allow us to sleep much better, right? And drowsiness. Levels in extracellular fluid activate other type of metabotropic receptors. Yeah. That's high levels. And adenosine and caffeine, they have similar chemical structure. However, they compete right at the binding sites. So there's this thing called A1 receptor. It inhibits glutamate release. So this is actually not desirable, yeah? It's functioning as an antagonist. And caffeine, so it blocks binding of adenosine A1 and A2A receptors. So in other words, caffeine is acting like an antagonist on these receptors. Right? What these things are, I will go into detail in our tutorial. So that's why after we feel coffee, since adenosine receptors are blocked by caffeine, right? We are kind of alert. We have much more energy. We don't feel tired. All right, quiz time. Serotonin is a or an? The answer is in a diagram. Okay. How many of you picked A? Hands up. Hands up. Hands up. Right. How many of you picked B? How many picked C? Yeah, right. Everybody got it right. So the answer is A, right? So the correct answer is A. Tyrosine serves as a precursor or building block, for trick question. This is a trick question. Yeah, literally you have to read the entire list of options. Right, so I will go from bottom up. How many of you pick E? How many of you pick D? How many of you pick C? Right, okay. Everybody got it. The answer is E, right? Dopamine, norepinephrine, epinephrine, right? There's three catecholamine. So neuropeptide and gasochemicals, that one I will, gasotransmitters, right? I will just go through it very, very quickly. Right. So I just really have enough time to talk about this oxytocin. So oxytocin is what we call the love hormone. So you guys can read, it's synthesized in the cell body, transmitted to the axon terminal. And so the number 3 bullet point number 3, all neuropeptides are metabotropic because they target metabotropic receptors. So this means neuropeptides are slow to, it produces slow reactions. Yeah, the slower to act produces slower reactions. And they act as neuromodulators and / or neurohormones, right? So in this case, oxytocin can act as both. So yeah, neuropeptide oxytocin, love and bonding hormone. Yeah, for lovers, for parents, for mothers giving birth, oxytocin is always secreted because it makes you feel good. Right? And there's this substance P for pain perception, memory cell growth. This really is for everybody's knowledge. And we also have this thing called endogenous morphines. Once we truncate it, we call them endorphins. They're useful for pain reduction, and they act on the same receptors as opioids, such as heroin, which we'll talk about in tutorial. And neuropeptides are involved in digestion. So we have this thing called insulin and cholecystokinin, right? Yeah. Very interesting word. It's from Greek. It means like move your gallbladder to release the bile into the small intestine. Yeah. So these are just two other interesting neuropeptides. Right. Okay, very quickly I'll talk about gastrotransmitters. So they transmit information from the postsynaptic to the presynaptic neurons. This is totally unintuitive, non-intuitive, the other way around. So instead of coming down, it goes up. And it breaks down quickly without enzymes. Another feature, right? So they dissolve in lipids because they are gaseous in nature, right? Nitric oxide, carbon monoxide, they are gases within our body. So they diffuse the membranes and interact with intracellular receptors to transmit information. There's no particles at all, right? I mean, the particles are just gaseous in nature. They're not like particles in the form of our neurotransmitters. Okay, so I talk about nitric oxide. So it's important for neurocommunication. It's important for maintenance of blood pressure. Important for erection. So if you want to write about erectile dysfunction, this is your chance, right? And it activates secondary messengers, right, like our G protein. Drug action in the synapse. So in the last 10 minutes, I will talk about this thing. Drug action in the synapse, which is highly useful for our tutorial. So, all these drugs, they can increase or decrease the activity of neurochemical. So in a nutshell, what an agonist do is that it enhances activity of a neurotransmitter, right, increase the excitatory or inhibitory effect. Okay? And all these effects, I have a table in our tutorial, 2 slides. I will go into them in detail. Extremely important. And antagonists basically do the reverse. They reduce the activity of a neurotransmitter, okay? Decrease this excitatory or inhibitory effect, okay? Increase degradation of neurotransmitter, blocking receptor site, reduce synaptic release. Yeah? Okay. Mechanism of drug action in the synapse. This is really for everybody to study before you come to the tutorial. Okay, I will test everybody on that again. So the first one is the nucleate synthesis of neurotransmitter. It may affect the amount available for release. Yeah. So there's a consumption of high carbohydrate foods, right, lead to the generation of tryptophan, right? You drink a lot of milk, and milk again is synthesized into serotonin. So based on this theory, if you drink a lot of milk, you should feel good. Yeah? And neurochemical storage. So the keyword is always vesicles. So do your drug interfere with the storage of these neurotransmitters within a vesicle? So we do have such drug, it's called reserpine, right? It's used for treating high blood pressure. It blocks such monoamine, right? Block serotonin, for instance, into the presynaptic vesicles. Yeah, so basically, you don't really have a lot of serotonin, in your vesicles. That's not very good. Yeah? But then again, that really regulates your high blood pressure. Yeah? So the last part would be the chemical release. Okay, modify neurochemical release by interfering with exocytosis. Right, that means the fusion of your vesicles with the membrane and then breaking apart to release the neurotransmitters. Or neurotransmitter reuptake, that's what I mentioned just now, right? So this autoreceptor involved in taking the neurotransmitters back. Okay, so before I go on, I just want to mention this drug, AMPT, right? So it interferes with catecholamine synthesis. So you won't get a lot of catecholamine, right? And that is an antagonist, right, for such catecholamine. Yeah. Again, it's kind of hard to pronounce this word, I have to admit. So synapse receptor effects, right? Agonist in synapse, so the drugs can mimic the action of the neurotransmitter, can emulate the action. Antagonist is usually, this is like one of the primary functions, so you will bind the receptor, you will compete for space with the particular neurochemical, and you will not really activate the receptor. Yeah. So in detail, we'll talk about it in our next tutorial. Okay? So nicotine and muscarinic are ACH agonists, they mimic ACH, cholinergic neurons, receptors. So number 2 is a toxic amazon plant, a curare, curare and snake venom. They block nicotine, so it leads to paralyzing effects. And we have sedatives, in contrast to stimulants, this really enhances GABA reception, a GABA type A receptor. So reuptake inhibitors, reuptake effects enzymatic degradation. So we have cocaine, we have amphetamine, that one I will go through during the tutorial, and we have retelling. I mentioned this now, for treating ADHD. So they block dopamine. In other words, the dopamine antagonist. SSRIs, okay, selective serotonin reuptake inhibitors, they block the reuptake of serotonin. As a result, you have a lot of serotonin in your synapses. And that will kind of, theoretically speaking, that will improve your mood. And we had this very interesting group of phosphates called organophosphates. So they come from our pesticides, and they can be involved with ACH breakdown. They affect the function of ACHE, right? Acetylcholinesterase, that breaks down ACH. All right, so that's a lot of stuff, right? And drug interactions at the cholinergic synapse, ACH. Take note of this. This is actually one of your activity, right? Actually, the first activity for your tutorial. Okay, so for those of you coming tomorrow and on Wednesday, right, take note of this. Right. Interesting fact to know, right? I'm not going into in detail, right? We're running short of time. Okay. So which drug is not an agonist? Okay, all right. This is, I know it's a bit hard. Okay, spider venom, nicotine, botulinum toxin, SSRI. Okay, let's go from the bottom. SSRI, anybody? Hands up. That means it's an antagonist of something. Nobody picked D, right? Okay. Let's go to C. Botulinum toxin, which is Botox. Once you have Botox, you get firmer cheeks. Yeah? How many of you pick Botox? Hands up, hands up. Yeah. Very good, very good. How many of you pick B? Nicotine. Nicotine is a stimulant, huh? If it's a stimulant, it's very unlikely to be antagonist, right? So the answer is C, right? Most of you got it, right? The Botox, right? So it's antagonist of ACH. Yeah. So drugs, I want to go through this a little bit quicker. So effects of drugs depend on administration of drugs. So we have 3 protective mechanisms, our blood-brain barrier, our liver enzymes, and we have this area called area prostrema in the medulla oblongata. I'm not too sure where it is, but it's good to know. Yeah, it's good to know. Right? This is something I know when I'm reading the text. However, it is influenced by individual differences, all these effects. So there's a few considerations: body weight, gender, and of course genetics. Right? Genetics. It's always genetics is an issue. So with some focus on genetics, we have this liver enzyme ALDH. Some people just call it ADH, like me, right? So we need to use it to metabolize alcohol, right? So in the literature, it's kind of widely known that Asian people in general do not really have this ALDH enzyme, so we get drunk much more easily compared to Westerners. And placebo effect, I'm sure a lot of you know this. I've learned this from research methods. So it's our user expectations, which are influencing drug effects, even that we can be using a vitamin pill. And so as a result, we need to use double-blind experiments. So both the participant and the experimenter do not really have some preconception, pre-knowledge of what the experiment entails. So in our textbook, we have this amazingly interesting diagram telling us that concentration of a drug in the blood supply it depends on the method of administration. So one is by smoking nicotine, the other is really by chewing it, ingesting it through our mouth. So the smoking part, right, you will produce a faster increase in blood nicotine. Right? Why is that so? Because you're inhaling nicotine. You're inhaling nicotinic substances. So that really diffuses directly into your cell and into your blood supply. And why does it diffuse away? Why does the slope goes down so rapidly? It is because that thing diffuses away. Since it's gaseous, it's highly mobile, it diffuses away. This is not surprising. So the chewing part perhaps is a bit non-intuitive because it's kind of ingested. My guess is that it actually sticks to your membrane, right? So everything is not taken up so fast by our cells, right? So that's why you kind of produce a highly sustained concentration in smoking. I'm not too sure where they actually, okay, this is from our blood, but which part of our body is the blood sampled from? I really have no idea. I suppose it's the brain, okay, I can be wrong. So tolerance and withdrawal, there's a lot of text, okay? But tolerance, I'm sure a lot of you actually know about this. So essentially it's about weaker drug effects because you keep on using the drug and that will lead you to administer more, right, of the same drug, not a different drug to produce the same effect, right? So caffeine is one very good example. And withdrawal is what happens, suddenly you just stop using the drug, right? So it gives you the effects that runs directly in opposition to the usual effects caused by the drug. Yeah, so it will cause behavioral effects, like for example, if you haven't been using caffeine for a very long time and then you suddenly stop it, you'll feel lethargic, you'll feel tired. Right? So the last bullet point is what I said, you'll feel more drowsy. Addiction, right? So it's the compulsive need to just keep on re-administering the drug despite having negative consequences. It is like OCD. It is an obsession, it is a compulsion, right? And our dopaminergic system is highly, highly involved, right? So we talk about nucleus accumbens, yeah. And the last part is just like changes in gene expression brought about by such reduced responses. Again, this is our gene times environment interaction. I know that some people are writing about this, so it's a very, very good topic to write on. Okay. And vaccination, okay, we all know what this is, vaccination, yeah? We've been through COVID, so just want to talk about the first bullet point. Basically, is that, the vaccine, the molecule, is a mimicry of the harmful molecule, the harmful substance. So it binds to the molecule of the problematic drug, to simulate the kind of effects of the drug, and then to make your immune system work harder, right? So the third point is what your author mentioned. Is this a good thing? That vaccination should all these people having high risk of addiction? This is an ethical question, right? Yeah. So the thing is that adolescents may just use more to achieve the same effect, right? Instead of just going for vaccination, let me just take more of the drug. And should you take more of the vaccine? Alright, thanks everyone. So I will continue into tutorial 2. Yeah, I know there's a lot of noise, everybody wants to leave, okay? So do take note, alright, of the exam, and for those of you who have difficulty, who think that you have difficulty, please come and speak to me right now.